So today we're going to talk about mitosis and the cell division cycle. And we probably will not get through all of the slides that I uploaded. So I'll pick up wherever we leave off on Friday when I am going to be talking about meiosis, which is kind of like mitosis on steroids, and that it's a little more complicated. So I'm going to talk partly about the mechanics of meiosis, of mitosis today, but also about how mitosis is controlled in a regulatory sense, what the control network is that takes cells into and out of mitosis and allows them to replicate and divide their genomes in an accurate way, which is totally essential for all cells and all organisms. So this is just a summary of some of the key topics. It's mostly from the textbook, but I modified a couple of things. These are sort of the take-home messages from at least the first part of the lecture, so just an outline, really. So the cell division cycle is what we call the mechanisms that enable cells to reproduce asexually. And in order to grow and to proliferate, cells have to replicate their DNA, which we've already talked sort of obliquely about. We haven't really gotten into the mechanism of DNA replication, but that happens in part of the cell cycle called S phase. And then they go through rounds of division called mitosis, where they separate their chromosomes. And they have to replicate exactly once each, before they divide, so that the genetic complement of every cell is the same. And there was enormous interest in the kind of late 70s to 80s in understanding what the mechanisms are that control the cell cycle and identifying the molecules that enable this to happen in a highly reproducible and controlled way. This table is not something you should memorize, but it's just to give you a sense of how long this cycle can take. It highlights some of the shortest cell cycles. I'll show you an example of Drosophila embryos, which have a very, very rapid early cell cycle. So they go through this whole process every 8 minutes in the very early stages of development. Conversely, mammalian cells can take a day or many days to go through division. Yeast cells, which are very simple, small cells, take about 90 minutes to go through a whole cycle under optimal conditions of rich medium and so forth. So it varies a lot. And I'm not going to talk about this today, but many cells sort of pause and stop dividing either for a long period or permanently, but they can then be induced to kind of re-enter the cell cycle under specific conditions. So let's talk about how this works. Okay, so this is a movie, which again, I'm sort of inviting disaster, but this is a classic movie showing phase contrast microscopy. These are chromosomes inside the nucleus, and you can see them again because the chromosomes have a refractive index that differs from the surrounding, and you can see that these are condensed chromosomes. So they're already in mitosis. They're getting ready to divide. And this movie kind of illustrates what happens during that process if it plays. So,
as this plant cell, taken from a lily, prepares for division, the chromosomes first condense.
I just realized that I probably did not click the share sound with the recording, so it may or may not record, but I'll upload it. Can you guys hear it? Okay.
Yeah.
I'll play it with the sound.
As this plant cell, taken from a lily, prepares for division, the chromosomes first condense. Next, the mitotic spindle lines them up in the center of the cell. At the metaphase to anaphase transition, the sister chromatids of every chromosome pair separate suddenly in striking synchrony. The chromosomes are pulled along the microtubules of the spindle to opposite ends of the cell. After chromosome separation, membrane vesicles line up in the center and fuse with each other to form the new plasma membranes that separate the two daughter cells. At telophase, the chromosomes decondense in the newly formed nuclei.
Okay, so that kind of illustrates how abrupt the transition from being together to separating, the metaphase to anaphase transition, as we call it, is. So, and these are still images from that video, again, showing the chromosomes in their condensed state, and then lining up the metaphase plate, and then this stage where they're separating, which is called anaphase, and then the growth of a new membrane between the divided chromosomes, which is cytokinesis. And this is a plant division. The book makes a little bit much of like the differences between plant and animal divisions. They're mostly similar, except that the way that the new plasma membrane is built in plants is somewhat different from animals, and I don't really care about that, but so I'm not going to get into that. Okay, so this is sort of my version of this whole process. And so the cell cycle is sort of divided into either 2 or 4 stages, depending on how you look at it. All 3 of these stages, which include S phase, the stage where the DNA is replicated, and 2 gap phases, G1 and G2. Together, these are called interphase. And then mitosis is the period during which the chromosomes are condensed and undergoing this division. And we talked early on in my lectures about how early cytologists, they saw chromosomes using a variety of techniques. And so they named the stages of mitosis based on what the chromosomes were doing in the microscope. And so they have specific stages or substages that are, it may seem hard to remember, but we'll talk a little bit about what happens in these different stages. But basically, prophase is the beginning of mitosis, when the chromosomes are condensed, but they haven't really started moving yet. Metaphase is defined as the point when they are lined up in the middle of the cell. That's meta, is middle. Ana is when they're moving apart. Telophase is kind of the end, when they are far apart and they're starting to decondense. And cytokinesis describes the formation of a new boundary between the 2 daughter cells. And these are the major events that happen in interphase and in mitosis, just as a summary. So again, S phase is the period during which DNA is replicated. In many cell cycles, there's a gap phase after mitosis, before the initiation of DNA synthesis, but not always. Sometimes they go directly from M phase into S phase. Similarly, often there's an extended pause after S phase before mitosis ensues. Okay, so obviously, it's, as I said before, it's really crucial that the cell control DNA synthesis, so it does it only once each cell cycle, and that the chromosomes are all fully attached and they all split in a very regulated way to make sure that every daughter cell gets exactly the right number of chromosomes. And errors in the cell cycle are, they're rare in normal cells, but they're much more frequent under certain circumstances, particularly in cancer cells, where the cells sort of lose their normal level of control. That's part of what makes them cancerous. And that can lead to the wrong number of chromosomes in different cells, which is called aneuploidy, which is a form of genomic instability and can lead to cell death, but can also lead to aberrant proliferation. Okay, as exiting M phase, the final exit. I'm not sure what Kirsten means there. Do we consider cytokinesis?
Yeah, someone's asking. Yeah, okay, yeah.
I see. So it's coupled to M phase, as I will talk about. I don't consider it part of mitosis per se, but I mean, that's semantics. If you called it part of mitosis, that would be fine. Okay, so I refer to mitosis really as the chromosome division cycle, and so I wanted to highlight some of the key things that happen during this cycle, and we'll get into these in a little more detail. So we've already talked about centrosomes, or microtubule organizing centers, which play an essential role in mitosis. So they're, as I mentioned, they're tethered to the nuclear envelope. Inside the nucleus, there are chromosomes, and I've sort of drawn them as squiggles here, but remember, you can't really see them as individual entities in interphase because they're decondensed. But in S phase, they replicate. So now there's 2 copies of each, and they are held together. The sisters, which are the 2 products of replication, are held together by what's called sister chromatid cohesion. And I will talk more about that either later today or on Friday, depending on how far we get. Okay, so mitosis ensues with two kind of major events. I mentioned that during S phase, the centrosomes or centrioles at the core of the centrosomes actually undergo replication too. We're not going to talk about the mechanism by which that happens, but one of the first things that happens in mitosis is the replicated centrosomes split into two, and they migrate along the nuclear envelope to opposite sides of the nucleus. And you can kind of see microtubules coming out here. That really varies depending on which kind of organism we're talking about. In some organisms, like budding yeast, these microtubules actually enter the nucleus, but in mammalian cells, before that happens, the nuclear envelope actually disappears. So that's kind of one point of divergence. Okay, so that's showing the nuclear envelope breaking down, which happens in as I said, animal cells, but not in every organism. And then the chromosomes start attaching to the microtubules that are emanating from these centrosomes, or now they're called spindle poles because there's two of them and they're making a spindle. So the chromosomes start moving around and trying to align, and they all get attached. They all get to the point where they're attached to both poles of the spindle, and then it's called metaphase. Before that, it's called prometaphase, and then it's called metaphase when they're all fully lined up. And at that point, the cell recognizes that all the chromosomes are properly attached, and it says, aha, it is now safe to divide. And that is when the metaphase to anaphase transition happens. And so as I'll talk about, the chromatids let go of each other, and then the chromosomes start moving towards the poles. And there's actually two phases of anaphase, one where the chromosomes are moving towards the poles, and one when the poles are moving apart. Those are called anaphase A and anaphase B, respectively. I am not going to, ask you to remember that, but the book talks about it, so I'm mentioning it. Okay, and then, as I said, telophase is the point at which the nuclei are now far apart. They start decondensing to go back into interphase. Okay, and then the whole thing starts again. So mitosis is a synonym for M phase, and they're, so you can use those interchangeably. And it includes the whole process of condensation, attachment, segregation. And these are just images of mammalian cells at different stages of mitosis. Yes, I will explain more about how the sisters let go of each other. And just to illustrate what my cartoons are trying to represent in kind of more real life. And these images all show DNA stained with a DNA dye, so you can see the chromosomes as they start to condense, and see the individual chromosomes, and then when they're lined up, that's metaphase. And the green here is microtubules. Okay, so I'm going to tell you a little bit about the history of how this was worked out. So there was a Nobel Prize awarded in 2001 to three of the major players in the discovery of molecular mechanisms that control the eukaryotic cell cycle. Lee Hartwell at University of Washington and Paul Nurse in, I think, Oxford in the UK, were both geneticists, and they used two different simple model systems, budding yeast and fission yeast, to carry out genetic screens that identified key components of the cell cycle. And I might talk a little bit more about how they did that, but I took that out for lack of time today. And then Tim Hunt was a biochemist who identified one of the key proteins that drives the cell cycle by accumulating and disappearing each cell cycle. Okay, and that's just what that says. But even before these three guys did their important work in the late 70s, early 80s, people had started investigating the cell cycle, and one person who didn't win the Nobel Prize but arguably should have was Yoshio Masui, who pioneered the use of this amazing frog egg system to study the cell cycle. And many people still use frog eggs and frog egg extracts to study the cell cycle, like Rebecca Heald at Berkeley and many others. So Yoshio Masui recognized that these eggs were a really nice system because you can get a lot of them. So this is a frog. It's about yay big. They're kind of big, ugly frogs called Xenopus laevis, and you can keep them in tanks, and there's a whole bunch of them in LSA somewhere. And you can inject the frog with progesterone, which induces it to lay tons of unfertilized eggs, which look like this, and they're very large. And they have two main advantages for studying cell cycle. One is that these frog eggs are easy to manipulate. You can actually like stick a needle into them and pull stuff out, and because they're so big, and also they're very large, and they go through what are called cleavage divisions, which is very common among animals, which means that the first 12 cell cycles, the frog egg doesn't grow at all. It just cleaves, and the cells get progressively smaller and smaller. So they start out as one giant cell and get smaller and smaller. And so each egg contains all of the components that are required to execute 12 division cycles. They only have to make a few things, as I'll show you, to actually execute all of these 12 cell cycles. There's one protein in particular, cyclin B, which I'll explain in a minute, that has to be made and destroyed each cell cycle. So they're really rich in protein, and I'm going to play this movie so you can see the cleavage cycles. So there's the first one, second one, and so forth. And after this, the frog egg has 2 to the 12th cells, which is like 4,096, I think. So that means that the original frog egg contained basically all the RNA and protein necessary to execute 12 cell cycles and to build, over 4,000 cells. So it's a very rich source of material. And one of the key experiments that Masui did is, was he showed that there was something in these eggs that could drive cells into mitosis. And I'll try to explain that a little bit more clearly. So when the eggs are laid, when they're first laid, they're arrested in M phase. It's actually a meiotic M phase rather than a mitotic M phase, but it turns out that the control of those division cycles is at the level of the regulatory proteins is not that different. And what Masui found is that you could get the eggs to go into interphase by treating them with something that, sorry, this is not interphase. This is interphase. You could get them to go into interphase by adding something to that causes the membrane to become permeable to calcium, and that would trigger them to go into interphase. Okay, so no, they're not 12. I'll come back to and answer that question a bit later. Okay, so what he found is that you could take some cytoplasm out of this M phase arrested oocyte and inject it into an interphase oocyte, and that would drive it into interphase. You would start to see spindles form and other signs that the frog egg was in M phase. And conversely, if you take cytoplasm from an interphase oocyte and inject it into an M phase oocyte, that does not put it into interphase. And also, if you take cytoplasm from an interphase cell into an interphase cell, it does not put it into M phase. Sorry, I didn't explain that very clearly. But the upshot is that there is something in the cytoplasm of M phase oocytes that can drive a non-M phase oocyte into M phase. So something kind of acts dominantly to drive things into M phase. So what is that stuff? So Masui called it mitosis promoting factor. And there was this huge kind of race to try to figure out what MPF actually is. So Tim Hunt, who did win the Nobel Prize, identified one of the key components of MPF. What Tim Hunt was doing was taking frog eggs, and also in this case sea urchin eggs, and injecting them with radioactive sulfur coupled to methionine. So this gets incorporated into new proteins. And so you can measure the synthesis of proteins on a blot where you look at these radioactive bands. And so the frog egg, as I said, or in this case, sea urchin egg, is chock full of most proteins that it needs. It makes some proteins gradually over time, like this one, ribonucleotide reductase, so that this line just shows kind of the synthesis of some proteins that the egg needs and makes sort of at a steady state over time. But they identified these other proteins, which they called cyclins, cyclin A and cyclin B are shown here, that both seem to go up and down and up and down. And the peak in their abundance correlated with specific states of the cell cycle, which they could monitor by actually looking at the eggs and seeing when they divided and when they formed spindles and so forth. And it turns out that this protein, cyclin B, is the key protein that has to be made and destroyed and made and destroyed to take cells into and out of mitosis. So every time a cell wants to go into mitosis, it has to make cyclin B, and every time it wants to separate its chromosomes and exit mitosis, it has to destroy cyclin B. And so what is cyclin B? So it turns out that this MPF activity that Masui initially identified is two things. It's cyclin B, which I just talked about, and what cyclin B does is it activates another protein, which is a cyclin-dependent kinase, or CDK. And the first CDK that was identified and shown to be essential for mitosis is called CDK1. It has other names that I listed here, which will probably just confuse you, but you may encounter other names for CDK1 based on the genetic screens in which they were identified in fission yeast or bunny yeast. So they have slightly different names in different organisms, which does not help you learn this stuff, but so be it. Okay, so this kinase called CDK1 drives cells into mitosis, and it becomes active when it's bound to a cyclin. And so the way that the cell cycle works is that cyclin B is made continuously. It's synthesized continuously. It's transcribed and translated continuously. And it reaches some kind of threshold level that then causes cells to have enough CDK activity, CDK1 activity, that pushes them into mitosis. So this illustrates that these proteins bind to each other, and the cyclin-dependent kinase, in this case CDK1, becomes active in part by binding to a cyclin protein. So this is a regulatory subunit, and this is the enzymatic subunit. Okay, so it gets a little bit more complicated because, as you might imagine, cells have to be sort of committed to doing specific things. They can't be wishy-washy. And so the cell cycle state alternates sharply between interphase and M phase. But cyclins are made kind of gradually because they're made by transcription and translation, and that takes time. So cyclin protein levels rise gradually over the entire course of interphase. But the MPF or CDK1 activity level does not exactly follow this rise in cyclin activity. Instead, there's a threshold effect where when the cell has enough cyclin, then it undergoes sort of a positive feedback loop that causes a sharp rise in MPF activity, and that drives the cell into mitosis. Okay, and then as I'll talk about in a couple of slides, there's an abrupt drop in cyclin protein levels, which is mediated by destruction. So there's an activity that I'll talk about called the anaphase promoting complex that turns on at a certain level of MPF activity, and that triggers the degradation of the cyclin protein, and that's how cells actually split their chromosomes and exit mitosis. Okay, and the book talks about something that I'm not going to get into in this lecture, which is that there are actually multiple different CDKs and different cyclins especially in mammalian cells that control different things that happen in the cell cycle. I am focusing exclusively on this M phase cyclin and M phase CDK. But there are others that are important for example, initiation of DNA synthesis to kick off S phase. So you can read more about that if you're interested. There's also a table that lists the different cyclins that are present in vertebrate cells and their different CDK partners. We're just focusing today on M phase and the cyclin and CDK that drive M phase entry and exit. So just focus on cyclin B and CDK1. But the similar regulation applies to all the different cyclins and CDKs in the cell. Okay, so I mentioned that the exit from mitosis occurs in an abrupt fashion, and it's driven by destruction of this key regulator cyclin B. And the way that this works is something that we've kind of touched on very tangentially in previous lectures, but we haven't really talked about the mechanism. So what happens is that as MPF levels get high, that triggers activation of this complex called the anaphase promoting complex. And there's a /C here because there's another APC in cells that is not the anaphase promoting complex. So this is just to differentiate them. Okay, so the anaphase promoting complex is what's called a ubiquitin ligase. And I don't know if you guys have learned about ubiquitin before, but ubiquitin is a marker that gets put onto other proteins and triggers them to be degraded. So we've talked about various kinds of post-translational modification, phosphorylation being probably the most common that we think about. Also talked about modification of histone proteins by histone methyltransferases and acetyltransferases and so forth. This is a different kind of post-translational modification called ubiquitination or ubiquitination, either, they're synonymous. And it's actually, ubiquitin is actually a very small protein. I don't remember how many amino acids it has, but some of those amino acids are lysines, and you can form chains of them by attaching the C terminus of one ubiquitin molecule to a lysine on another ubiquitin molecule. And this one is attached to a lysine, in this case on cyclin B. So once this gets activated, it quickly builds a chain of ubiquitin on the cyclin B, and that triggers destruction of cyclin by this machine that eats up protein called the proteasome. Okay, and then the CDK, the kinase, becomes rapidly inactivated because it no longer has its cyclin partner. To make matters a little more complicated and harder to remember, CDK activity is not, sorry, I'm putting my cursor on the wrong screen. CDK activity is not purely regulated by binding to cyclin. It is also regulated by other mechanisms involving phosphorylation. And the reason for that may seem kind of like, why do cells bother with this? But it enables them to regulate the activity of CDK in response to more inputs, as I'll show you in a couple of slides. So just because CDK is bound to cyclin, that is not sufficient to make it active. There are also inhibitory phosphates that are put onto CDKs. In the case of the mitotic CDK, those phosphates are put on by a kinase called WEE1. And you might imagine where it got its name, that it was found because cells that lack this inhibitory kinase, they enter mitosis too early, so they make very small cells. So they're WEE. So WEE1 puts these phosphates onto CDK, and then when the cell is confident that everything is properly set up for it to go through mitosis, then it removes those phosphates using a phosphatase called CBC25. So this is a case where phosphates inactivate this kinase, this enzyme, and removing the phosphates activates it. Okay, so when those phosphates get removed, now you have active M-phase CDK, and the cell can go through mitosis. So this is an example of a positive feedback loop where you have, so once the phosphatase becomes active, or once a little bit of the phosphatase becomes active and activates the CDK, that also triggers more activation of the phosphatase and more CDK activation. So that's how you get this very sharp rise in CDK activity that takes the cell abruptly from interphase into mitosis. Okay, so that is part of why you have this like threshold-like behavior. Once you get to a threshold level of cyclin protein, that positive feedback loop starts up and you get this sharp rise in MPF activity to drive cells into mitosis. Okay, and then I mentioned this also that at a certain point, when cells are in mitosis and then the chromosomes have properly attached to the spindle and everything's good to go, that enables the activation of the anaphase promoting complex, which drives the degradation of cyclin and exit from mitosis into the next interphase. Okay, so this is like the textbook's version of my little cell cycle diagram, but I just wanted to make the point here that this cycle of synthesis and destruction acts kind of like a ratchet, so that the cell cycle can really only go in one direction. It can't slip back because the cyclins are destroyed as it goes around the cycle, and so that keeps it going in a unidirectional fashion. Okay, so we've talked a little bit about the complexity of this regulation. And one reason that the regulation is complicated is that the cell uses a lot of information to make sure that everything is okay before it goes on to the next stage in the cell cycle. And so it uses mechanisms that are collectively called checkpoints to monitor different things that are supposed to happen during this whole cycle. And it will arrest the cell cycle at different points if things are not where they should be. So in particular, DNA damage, which occurs frequently as just a byproduct of DNA replication, but can also, occur spontaneously by radiation or oxidative damage or other things. DNA damage can arrest the cell cycle in a number of different places. It can block M phase entry, but it can also block even S phase entry and progression through S phase. So there's a very robust system that recognizes DNA damage that we're not going to talk about in any detail, but it can arrest the cell cycle. And I will talk a little bit more about this. So I mentioned that the cell is checking that all the chromosomes are properly attached to the spindle before it executes the signal to separate the sister chromatids and allow them to divide. And that's really crucial because if the cell tried to divide and there was an unattached chromosome, then this chromosome would go to one of the two daughter cells and they would become aneuploid, which is extremely destructive. So spindle assembly defects, what's called the spindle assembly checkpoint monitors both the integrity of the spindle and the attachment of the chromosomes to the spindle. And if it's not satisfied, that blocks the metaphase to anaphase transition. It blocks it actually by preventing activation of the anaphase promoting complex. There's also in some yeast cells additional checkpoints that monitor, like if the spindle is in the middle of the cell and so forth. So that's another thing that can arrest the cell cycle at mitotic exit. And part of why I'm bringing this up is because one of the things that happens in cancer is that one or more of these checkpoint mechanisms can become compromised by mutation or loss of one of the key components of the checkpoint. And one of the main things that happens in cancer cells, in roughly half of cancer cells, this gene called P53, which is probably the most popular gene to study in all of biology, and yet it has a super lame name. It's just like peptide 53 kilodaltons, basically. This is a protein that is not strictly required by cells, but it is required for them to execute the cell cycle faithfully because it is an important component of DNA damage checkpoints. So I mentioned that cells have this ability to detect broken DNA. And one of the things that happens if the cell detects this is that there are kinases that get activated that we're not going to talk about. And one of the targets of those kinases is the P53 protein. And if it gets phosphorylated, P53 acts like a transcription factor and goes in and leads to the synthesis of other genes with very unimaginative names, one of which is this P21 protein, which is a CDK inhibitor. And so, sorry, I'm laughing at Kirsten's comment on the chat. So P53, this isn't the only thing that it does, but it is required for arresting the cell cycle in response to DNA damage. And it is the most frequently mutated gene in cancer cells, even more than RAS, which I talked about before. Okay, and this just shows, you do not have to memorize this, but this just shows a little bit more detail about the different checkpoints that I mentioned in my previous slide. And notice that P53 is involved in quite a few of these pathways that lead to cell cycle arrest, especially in response to DNA damage at different stages. Okay, I cannot see my clock, so I don't know what time it is. Can someone tell me what time it is? Okay, 9:48, so we have like 12 more minutes. So I'm gonna go ahead and talk a little bit about the mechanisms of chromosome segregation during mitosis. And this is just a summary of what I'm gonna say. So I already mentioned this first part, that the centrosomes duplicate during S phase, and in mitosis they migrate to opposite sides of the nucleus to form the poles of the mitotic spindle. And then I'll talk a little bit more about these other mechanics of chromosome segregation over the next few slides. Okay, I showed this slide in my last lecture on microtubules, just showing it again to highlight how the major change in microtubule organization between interphase and mitosis. And I talked a little bit about microtubule-associated proteins last lecture, and so a lot of those microtubule-associated proteins are targets of like CDK1 and other kinases that are themselves activated by CDK1. So as cells enter mitosis, the whole behavior of microtubules changes dramatically, in addition to the organization of the microtubule organizing centers, and that leads to the formation of the spindle. And just to kind of give you a sense of how dramatic this is, so this is a fly embryo, and again, these mitoses happen really, really fast, like every 8 minutes for the first several cell cycles. So it's looping, but you're basically seeing the chromosomes condense. In green here, they're labeled with a GFP tagged histone protein. And in this case, the microtubules are red because they are fused to an RFP, a red fluorescent protein. And you can hopefully see the signal that is actually present throughout the whole embryo. So this embryo is something called the syncytium. It has nuclei, but all the cells are basically one giant cell. And so all of these nuclei are getting they're seeing the same rise in CDK activity. And so it's triggering mitosis synchronously over the whole embryo. And after the first, I think, 14 cell cycles, these cells will form more complete cells and will behave more independently. They won't all divide at exactly the same time. Okay, so this shows spindle assembly, and so we've alluded a little bit before to kinetochores. The kinetochores are the structure that enable chromosomes to attach to microtubules and are essential for their division in mitosis and meiosis. So this is not a very nice picture, but it just shows a chromosome, and these dots here are fluorescent, showing the location of a kinetochore protein. And I know these terms are a little bit confusing, but chromosomes have this special region called a centromere, which has special histone variants associated with it that mark it. And what happens in mitosis is this structure called a kinetochore starts to assemble at the centromere. And this is a very simplistic view of the kinetochore. So the kinetochore has many different proteins and forms like this layered structure. And some of the proteins directly bind to microtubules and enable the chromosomes to actually move with a microtubule as the microtubule is depolymerizing from its plus end. So remember, the minus end of the microtubule is over at the spindle pole. The plus end is contacting the kinetochore, and as that microtubule shrinks, the chromosome actually moves with it. So this structure has to be able to link the chromosome to a shrinking or growing microtubule, which is kind of an interesting engineering feat. And this just shows another view of the spindle and the kinetochores, showing that there are microtubules that connect each. So in this case, it's showing that each chromosome is a pair of sisters that are still held together. And so the kinetochores are actually facing outward toward the spindle poles. And each of them forms connections to the spindle poles through microtubules. So these blue microtubules are what are called kinetochore microtubules. I mentioned in the previous lecture that there are end-binding proteins that stabilize microtubules, and there are end-binding proteins at the kinetochore that stabilize microtubules when they make contact with the kinetochore. The spindle is a little bit more complicated. It also has these overlapping microtubules that are not bound to kinetochores that the book calls interpolar microtubules. And those play an important role in cytokinesis. They actually specify where the cell will divide, and they physically couple the process of cytokinesis or cell division to the organization of the spindle. And that makes sure that the cell divides in a plane such that there's a set of chromosomes on each side of the division plane. So Max asks, is the kinetochore a fundamental part of the centromere or just something added on top of the centromere? So the centromere you can think of as the chromosomal region that specifies where the kinetochore is going to assemble. So the centromere is kind of the chromatin that underlies the kinetochore, and the kinetochore assembles so in some organisms there's kind of some kinetochore components there throughout the cell cycle. In other organisms, like the whole kinetochore basically assembles each mitosis. So the kinetochore is physically associated with the centromeric region of the chromosome, but it's sort of built out from the chromosome. Okay. Yeah, I know these are kind of confusing terms, but so be it. Okay, and I've also mentioned that spindles, most spindles have these additional microtubules that kind of flare out away from the spindle pole in the opposite direction from the chromosomes. Those are called astral microtubules, and they actually make contact with the cell cortex, and they help to position the spindle, that they balance the forces so that the spindle winds up kind of in the middle of the dividing cell. So they help to position the spindle properly. Okay, and then, so we've talked about most of this already, that, you know, this is the organization of the spindle, and then this shows what happens at the metaphase to anaphase transition, where the bonds that hold the sister chromatids together, which we haven't really talked about yet, get cleaved, and that enables the sisters to come apart. Astral because they look like stars, as Kirsten said on the chat. This shows another view of that. So you can see here, this is metaphase with a beautiful spindle. You can't really see the spindle poles here. This is kind of a different view of it. And the sisters are still held together kind of along their whole lengths. And then there's an abrupt transition, and what's called cohesion that holds the sisters together gets cleaved very quickly, and that enables all the chromatids to split from their partners and start moving toward opposite poles. So it turns out that this splitting of the sister chromatids is another key function of the anaphase promoting complex. So I talked about the anaphase promoting complex being this ubiquitin ligase that ubiquitinates cyclin B and thus enables mitotic exit. But it also it also triggers destruction of another protein called securin. And this is a little bit complicated, but hopefully you'll see the logic of it, and that'll help you remember it. So cohesion is shown here as these rings that hold together the sisters. This is very much not to scale. These cohesion rings are much, much smaller than they're pictured here, but there are these proteins that hold together, that pin together the sister chromatids in many, many places along their length. And one component of this complex gets cleaved by proteolysis. It gets digested. And that's what causes these rings to basically come off the chromosome and enable the sisters to separate. And normally that does not happen because of this protein called securin. So the rings actually get cleaved not by the proteasome, but by another protein cleavage machine called separase. It's actually just one protein that cleaves one of these cohesin proteins. So normally there's an inhibitor called securin that prevents that from happening. But when the anaphase promoting complex becomes active, one of the proteins it targets is the securin protein. That gets digested. Now separase becomes active and cleaves the cohesin complex, or one of its components, to enable sisters to separate. And I'm going to just show a different view of that here. This is maybe a little bit more to scale. So this just shows the individual DNA strands, and this is probably more to scale. The cohesins are thought to form rings. This view is being challenged now. The idea that the way cohesins work is actually to encircle both of the sister chromatids. It's more likely that they actually work in pairs to hold sisters together. So that don't take this too literally, but the idea is that these ring-like cohesin complexes hold together the sister chromatids. And I'll just remind you that we talked about cohesins before, because in addition to holding the sisters together, they also form these loop structures that we talked about when I talked about nuclear architecture and chromosome organization. They form these loop structures that are thought to be different domains within the chromosomes. And yeah, I'm not too happy with any one of these images, but I'm showing you like a variety of them to give you an idea of how people conceptualize this. So these complexes, these ring complexes, the condensin complexes, actually have more subunits than were shown in those previous slides. They have these two very large subunits called SMC proteins that form most of the bulk of these complexes. But then these two proteins, which are very similar to each other, are held together in a ring by a third protein called a glycine, which comes from the Greek word for clasp. So this glycine is actually the thing that gets cleaved by separase. And that's what's shown here, is that separase is inhibited by securin. When securin gets destroyed by ubiquitin-mediated proteolysis, that enables separase to cleave the glycine subunit. There it is, eating, it's a little Pac-Man eating the glycine subunit, and that's what releases these cohesins from the chromosomes and allows them to separate. So Naomi asks a really excellent question, which is, how is cohesin how chromosomes condense for mitosis? Not quite. So I decided not to talk about this today, and I may bring it up on Friday. It turns out that there's another complex that looks very much like this, but is called a condensin. Actually, there's two condensin complexes, which are evolutionarily related to cohesins. They are not, condensins load onto the proteins during mitosis. So during, prophase, at the beginning of mitosis, and they start condensing the chromosomes. And exactly how they do that is still not understood. But both of these complexes, cohesins and condensins, probably act like motors. One thing we do not understand at all is how cohesin and condensin have sort of different ways of packaging chromatin, so that the cohesin makes loops and holds sisters together, and the condensin causes chromosomes to become much more compact. Anyway, excellent question, and I just didn't feel like we had time to go into it. Okay, so, and I mentioned this earlier, that one of the functions of these spindle microtubules is to define the division plane of the cell. So here's the spindle, the chromosomes are all lined up along the spindle, and the spindle actually recruits a G protein that regulates assembly of this actin ring at roughly the midpoint of the spindle, and that's what triggers the cytokinesis to happen right at the point between the two halves of the spindle. So you wind up with one set of chromosomes on each side. And this is just showing cytokinesis, in this case, I think in an animal cell and probably an oocyte. And so this cleavage furrow, as it's often called, is made by a belt of actin filaments that kind of underlie the membrane, and then they are, the action of myosin causes those actin rings to shrink down and pinch off the cell into two new cells. And that's just shown here. So here's the contractile ring of actin forming at the spindle midzone. And here is a dividing cell stained for actin or myosin. And so this kind of contracting belt or ring is made of actin and myosin. And I think that's all I had. Oh yeah, I'll mention just one additional thing. So you might imagine that many things in the cell have to get rebuilt each cell cycle, and that is true. And one of the things that in many cells gets destroyed and rebuilt each cell cycle is the nuclear envelope. And so I mentioned that this is not true in all cells. Like in yeast, for example, the nuclear envelope actually remains intact during mitosis, but in animal cells, it gets taken apart and put back together. And that's just what's shown in this movie, is that you can see the components of the nuclear envelope. I think here it's a lamin protein that's stained in green, that they, as the cell is dividing, those proteins go into the cytoplasm, they move away from the nuclear envelope, and then they have to get put back very rapidly during telophase at the end of the next cell cycle. Sorry, to initiate the next cell cycle. And that's what's shown here. So these, many of the nuclear pore proteins and also the lamin proteins are targets of CDK1 and other kinases that are highly active during mitosis, and that's what drives the disassembly of the nuclear envelope. And then they get dephosphorylated as the cell re-enters the next cell cycle, and they, in a very orderly way, reform around the chromosomes. And it says, hey, Abby, it's past 10, so I will shut up, and I will be happy to take questions from anyone who wants to stay on for a bit. And okay, should we know all the specific pathways from the end of lecture 6? Friday, yeah, so lecture 6 was, we talked about myosin in the previous lectures. So I only talked tangentially about myosin because I didn't really talk very much about actin in Monday's lecture because I wanted to cover microtubules in more detail and we didn't have time. But myosin is the major class of motors that acts on actin filaments. And so no, I'm not gonna ask you any details about how myosin works because I didn't talk about it, but I did mention today that it's part of the contractile ring. Okay, the more specific RTK pathways. I focus more on the G protein coupled receptors. I mean, the quiz is going to be open book, open notes. So, I mean, you don't have to memorize this stuff, but you should understand the concepts that I talked about. And like I said, I'm not big on memorization, but on the other hand, the GSIs are mostly responsible for writing the quiz. So you should probably ask your GSI these questions. Anyone else? We didn't have any Zoom crashes today. That's a plus. I just had trouble signing in. Did the GSIs want to say anything else about the quiz right now, or do you want to just do that through B courses?
We're finalizing it today, so I don't think we have any. Anything up to Friday is fair game. If Dr. Dernberg covered it in lecture.
Okay. Can I elaborate more on M phases? Like specifically what? Like what are the subphases of mitosis? I mean, yeah, I'm not sure what the question is about, but as I mentioned, so mitosis was first described by people looking at chromosomes by microscopy. And so the different stages of mitosis were really broken down based on the appearance of the chromosomes at different times. Yeah, so, you know, so in prophase, they're condensed, but the nuclear envelope is still intact. Prometaphase is when they have initiated attachment to the spindle, but are not yet fully lined up. Metaphase is when they're fully attached. Anaphase is when they're moving apart, and telophase is when they have moved apart and are starting to decondense again. Like prophase to telophase versus cytokinesis. So cytokinesis is coupled to telophase. I think the state, I guess it initiates during anaphase and completes during telophase. So cytokinesis is the cell division process that is coupled to mitosis. But I distinguished it from mitosis because it's really defined by stuff that is not chromosomal, whereas these different substages of mitosis are largely defined by what's happening with the chromosomes. All right, everyone.
More questions.
Could I repeat what I said about MPF activity having a sharp rise versus cyclin gradually increasing? I'll just show you that slide again to illustrate it. So, right. So I mentioned that cyclin is synthesized continuously during actually the whole cell cycle. So you can imagine that the rise in cyclin protein levels is fairly constant, happens at a constant rate. But the activation of MPF is much more abrupt. And it has to be because the cell doesn't want to be kind of easing into mitosis. It wants to go into mitosis in a very committed way. And I talked about the fact that there's this additional level of regulation of MPF or CDK activity mediated by phosphorylation and dephosphorylation, partly because that's a major mechanism that causes this threshold-like behavior in the kinase activity, that you get a little bit of activation through the rise in cyclin levels, but then there's this positive feedback that once you cross this threshold, then the CDK activity, that's what's shown here, then that low level of CDK activity starts to feed back to activate more of the CDC25 phosphatase, which activates more of the kinase, the CDK kinase. And so you get that's this kind of positive feedback in biology is how you get very abrupt changes in activity. And so that's responsible for the fact that the level of MPF activity is not linearly related to the cyclin protein levels. It's related in a more complex way. Okay, if you have questions, related to the stuff that's on the big quiz, you can contact me by email. I think Piazza is the best way because chances are if you have a question, others have it as well. So I will make sure to answer any questions that are asked on Piazza, as quickly as I can over the next couple days. Well, I guess till tomorrow at noon. And then we'll probably inactivate Piazza again for the big quiz so that you guys are not tempted to communicate that way. All right. See you all on Friday. Good luck with the quiz. And